Children, next we will discuss about the vectors which are used for cloning genes in plants and animals. So, some common vectors that are there like agrobacterium, tumefaciens, which is responsible for cloning gene in plants. And next, that are retroviruses which are used for cloning gene in animals. Now first we will discuss about the Agrobacterium tumefaciens. Agrobacterium tumefaciens, this bacteria, they contain a plasmid, and this plasmid, it is known as Ti plasmid. This is known as Ti plasmid, and this plasmid, it infects the dicot plants. And cause crown gall disease. Which disease? Crown gall disease. Now what is this crown gall disease? In case of crown gall disease, it induces the tumor. Okay, means it induces the tumor growth. And how this tumorous growth, it occurs in the plants because of this Ti plasmid. Because in the Ti plasmid, suppose this is the Ti plasmid, in this Ti plasmid, one region is there. This is known as pDNA. This is known as pDNA region. And this region, it transfers into the plants. And this tDNA region, it is responsible for causing the tumorous growth in the plants, and they cause crown gall disease. Okay, let's see again. Agrobacterium tumefacin, it contains the Ti plasmid, and this Ti plasmid, it contains the tDNA region, and this tDNA region, it infects the plants, it easily transforms into the plant cells, and then it induces the tumorous growth, and this results in the crown gall disease. Now next, that are retroviruses. Retroviruses, they are responsible for converting the normal cells into the cancerous cells. In the animals. So we can use the Ti plasmid as well as the retroviruses as vector. But how we can use these viruses as well as the Ti plasmid as vector, as they contain the tumor causing genes. If suppose these genes which are responsible for causing the tumor or which show the cancerous growth, it can be removed, and in place of that gene the foreign DNA that is inserted, then they are known as the harmless vector, or you can say disarmed vector. Disarmed means that to convert the harmful plasmid or the viruses into the harmless condition, harmless vector. This is known as disarmed. Okay, making the vector disarmed means removing the genes which are responsible for causing the tumor or which are responsible for causing the cancer. Suppose this is the Ti plasmid. If suppose this tDNA region that is removed, and in place of this tDNA, the foreign gene or the foreign DNA, it is inserted. Now this Ti plasmid, it will become disarmed. It will become disarmed Ti plasmid. In this manner, this Ti plasmid, it is used for transformation of the foreign DNA into the host plant cells for producing the desired characters. Now in this case also, the retroviruses having the DNA, if suppose the DNA part which is responsible for converting the normal cells into the cancerous cells, if that part that is removed, and in place of that part if foreign DNA that is introduced, now it also become disarmed retrovirus. Now they don't have the disease causing gene. In place of that, they have the desired DNA. And then they can be transformed into the host cells for producing the desired characters.